Assalamualaikum everyone. My name is Maheen Sohail and I'm from Department of Biotechnology and Genetic Engineering, Kohat University of Science and Technology. I'm here to present about my MPhil research, which is the folic acid production and characterization of folic acid producing bacterial isolates from traditional food samples. Here's the outline of my presentation, which include introduction, methodology, results, conclusion, and references. So first of all, what is folic acid? Folic acid is actually a vitamin B9, which is a water-soluble vitamin, and it is a cofactor in several metabolic pathways, which is the conversion of amino acid and nucleotide synthesis in healthy and proliferating cells. So folate is actually like in variety of research studies have documented the effects of folic acid deficiency, the folate deficiency on birth defect. It is the folate deficiency that has been considered a crucial factor in neural tube defects. So as I have used the word folic acid and folate both words. So let's differentiate in between them. Well, folate actually refers to all forms of vitamin B9. All forms included folic acid, tetrahydrofolate, 5-methyl tetrahydrofolate, and a 5-formyl tetrahydrofolate. So why is it necessary to identify the folate, to quantify the to isolate or characterize the folate from bacterial sources? Well, humans and mammals can't synthesize folate and folic acid, so they must have to obtain it from the exogenous dietary sources, which is the vegetables, milk, and fermented dairy products. What are the objectives of my research? As I have mentioned earlier, that um, humans can't synthesize folate. So like we have to obtain it from diet. So here I'm using the some traditional food samples, which includes the milk and yogurt samples. Yogurt is actually a fermented food sample to check out the bacteria, the folic acid producing bacteria, how much they are folate producers, and and if they are producing folate, then how much amount they are producing that are required for human. Well, from the European Union conservation, they shows that a normal human being daily required has to require a folic acid amount which is the 400 microgram. So like for a human, for a single human, the daily intake of folic acid is 400 microgram. So here, what are the objectives? The objective is to isolation and characterization of folic acid producing bacteria from food sample by using the biochemical and molecular approaches. Next step is after the isolation and molecular analysis, biochemical and molecular analysis, we have to quantify the folic acid production from those bacteria, from those specific folic acid producing bacteria at different growth stages. Well, at different growth stages means here that like in which phase as bacteria grows in fourth phases like lag phase, log phase, and stationary phase, and the end one is decline phase. So at which phase these bacteria are producing folic acid, and how to quantify these, uh, how to quantify folic acid production of these bacterial isolates. So for quantification, we are using two processes and we will discuss it in methodology section. Next here is the methodology. The methodology includes the sample collection, and sample is collected from different dairy farms of cohort. And after the bacterial isolation, we have analyzed those bacteria on the basis of their morphology, plus identification was done through biochemical analysis. After that, molecular characterization, which includes the DNA isolation and their PCR and sequencing. The last step is the quantification of folic acid. So here are the results. Okay, let me give you some kind of examples, like how do we have done morphological analysis, or how do we isolate bacteria first. Firstly, so for bacterial isolation, we have sera diluted our milk sample plus the yogurt sample, and after the serial dilution, we have growed the bacteria. Well, we don't know what kind of bacteria they are, so we are growing it on the MRS agar plates. So MRS specifically only grows the lactobacilli. Plus, in the presence of when we add the calcium carbonate in the media and keep it at 37 degrees centigrade for 48 hours, so the bacterial colonies will be was isolated randomly, which forms a clear zones around itself by dissolving calcium carbonate. Well, those bacteria was selected which forms a clear zone around itself by absorbing the calcium carbonate. So after selection of those bacteria which form these zones around calcium carbonate, we culture them on pure plates, and after that preserve them in 20% temperature at -80 degrees centigrade. For the morphological and biochemical identification, we have used different types of tests. We will discuss it in the results section, and quantification was done to and quantification was done through two processes, which are UV spectrophotometric analysis and microbiological analysis. So first here is the results are the collection of food sample was collected from different dairy farms in Kohat, and sample was suspended in saline solution, and serial dilutions were made. As we have discussed it earlier, that we have firstly made the serial dilution of the samples, and after that we grow them in agar plates that have been mixed with 1% of calcium carbonate, and we just incubate it at 7 degrees centigrade for 48 hours. And those bacterial colonies were selected which formed zones around itself by dissolving calcium carbonate. So the question arises why they are forming the calcium carbonate, why they are forming the zones around itself by dissolving the calcium carbonate. Well, calcium carbonate is a kind of substrate for them. When the bacteria produce folate, any kind of folate, so it absorbs the calcium carbonate, while bacteria form clear zones around it by dissolving calcium carbonate. So bacteria, calcium carbonate is acting as a substrate, and when bacteria produce folic acid at its specific growth stage, so this folic acid actually absorbs the calcium carbonate, a reaction occurs, and they repel, and they just like repel the growth of bacteria. Next step is different tests was carried out to check about the morphology of these isolated bacteria. So here are the results of biochemical analysis, morphological and biochemical analysis. So when bacteria was analyzed to drug staining, well, different six bacteria were isolated, and the four bacteria were lactose, like gram stain positive, gram positive bacteria, and two strains were gram negative. All bacteria strains were catalase, the positive catalase test, five bacteria isolated to a positive oxidase test, and one isolated to E negative test. While motility of the all isolates were negative, while only three isolates produced were the positive for lactose fermentation, and three isolates were negative for lactose fermentation. Next step is the MRS. These are the MRS results of the four isolates that produce a zone in presence of calcium carbonate. So this is the zone. Next is the bacterial isolates. Okay. So next experiment was designed just to check out the activity of those selected bacteria in the presence of environmental stress. Environmental stress includes the temperature, different temperatures. Bacteria were grown on different temperatures to tell their activity. Next was the bacteria were grown on under different pH, under different NaCl concentration plus different concentration of ethanol. So here are the results of bacterial isolate tolerance with check on different temperatures. And the graph was made against their ODs, and there are four bacteria, and the color different colors is showing the different temperature. So all of the bacterial isolate, like they gave the best result on 37 degrees centigrade, which is, and the OD they have given is 2.7 and 2.0, like it's the maximum OD. Next is the figure A and B is showing the results of four isolates under different environmental state, that is the NaCl and ethanol. So the first one is showing the figure A is showing the bacterial growth rate, survival rate, which is actually under the different NaCl concentrations. So the bacteria one, which is actually isolated from a yogurt sample, it is showing a good result and in positive growth, like 80% growth rate at 3% of NaCl concentration, and around about 75% growth rate at 6% NaCl solution, and um, around about 55% survival rate at 8% NaCl concentration. While for ethanol, the bacteria were like they have a kind of same results, just because ethanol is actually, they actually kills the bacteria. So at the 10, 5% and 5% NaCl ethanol concentration, sorry, 5% ethanol concentration, so the bacteria were like they stopped growth, and at the 10% NaCl concentration, they just became linearized. Like the minimum growth, they have shown a minimum growth on 10% in the presence of 10% in ethanol concentration. Here is the survival rate of these bacterial isolate under different pH. So the bacteria, well, at the pH 2 and 3, they have shown a little less growth, around, that is actually 50% growth rate. And at the pH 4, they have shown a growth rate or survival rate of 55%, while at the, while they have shown the, while the bacterial isolates have shown a maximum growth at 5 and 6 pH. Next here is the calibration curve, which is actually drawn in between. Well, the blue line is showing the concentration of control sample. Control sample means like it's the calibration curve as standard sample was used. 5-methyl tetrahydrofolate standard sample will different concentration of that sample was made, of that standard solution was made, and their absorbance was checked on 282 nanometer. Well, 282 nanometer is used for folic acid detection, and a calibration curve was drawn. And the four bacterial isolates have shown the concentration of 1 to 4 microgram concentration of folic acid. So, like they have shown a maximum production of folic acid. The rest of the results are awaited. So from this experimentation, we have concluded that from the analysis of UV experimentation, we have concluded that a normal that amount of that folic acid is produced and broken and milk sample and is present in amounts that is required by a normal person in their daily life, which is actually a 400 microgram. So yes, through and we have detected this through UV analysis, and we have quantified the folic acid produced in bacteria isolated from food sample. So we have developed our own methodology of UV analysis, and that will be discussed further in research, in pieces, and in the research paper. There are the references that I have used to make this presentation. And thank you so much for your attention.